Good afternoon, everyone. Welcome to today's webinar, Genomic Integration of the SARS-CoV-2 virus and the Potential Relevance for the Course of COVID-19. I am Kip Weisskopf, a Valhalla Fellow here at Whitehead Institute, where my lab studies how immune cells called macrophages can be used to fight cancer in the body. I'm excited to be your host and moderator today. Today's webinar is part of a series of talks that we've organized to share Whitehead Institute science and research with the wider community. In a few moments, we'll hear from our presenter, Whitehead Institute founding member, Rudolf Jaenisch. First, I would like to let you know that this webinar is being recorded for future viewing. Also, we will be answering questions at the end of today's presentation, but you can submit a question at any time using the Q&A box at the bottom of your screen. With that, I'd like to introduce our speaker today, Rudolf Jaenisch. Rudolf Jaenisch received his doctorate in medicine from the University of Munich in 1967 in the lab of German phage researcher P.H. Hofschneider. Before coming to Whitehead, he was head of the Department of Tumor Virology at the Heinrich Pett Institute at the University of Hamburg. Jaenisch has co-authored more than 500 research papers and has received numerous prizes and recognitions, including an appointment to the National Academy of Sciences in 2003. At Whitehead Institute, his lab studies the genetic and epigenetic basis of diseases such as Parkinson's, Alzheimer's, autism, diabetes, and cancer, among others. This work has led to major advances in the understanding of embryonic stem cells and induced pluripotent stem cells, or iPS cells, which appear identical to embryonic stem cells, but can be created from an adult cell without using an egg. Jaenisch and his lab have focused on creating new ways to model human diseases in animals. In one project, the lab created chimeric mice with human cells in their nervous systems in order to better recreate the conditions under which tumors form in the human childhood cancer neuroblastoma. Today, Rudolf's presentation will highlight his most recent work on the complex biology of SARS-CoV-2, including the stages of SARS-CoV-2 infection and some other puzzling observations. These include the recurrence of PCR-positive tests in patients that have cleared all infectious virus and the long-term complications of the infection, also known as long COVID. He will discuss his lab's evidence for genomic integration of SARS-CoV-2 sequences in cultured cells as well as in patients. One question he will answer is whether vaccine RNA can also integrate into the genome, and he will discuss his lab's preliminary results suggesting that vaccine RNA does not in fact integrate. Rudolf, welcome and thank you, and the virtual stage is all yours.
Well, thank you very much, Kip, for this introduction, and good afternoon for everybody. So I am really very excited to be part of this series, and I share my screen. Okay, there we are. So I'm talking about SARS-CoV-2, and it's really a more recent line of research in my laboratory, but it's of great interest to us. And I just want to disclose some associations they have with companies. So I want to put SARS-CoV-2 in historical context, the human history and pandemics. And it's really quite a big history. The plague in 500 after Christ killed the equivalent of half the European population. Bubonic plague was really very serious, and again another one there, the cholera. The 1918 influenza pandemic killed 20 to 50 million people. The HIV pandemic only last century killed quite a lot of people. And now we have COVID-19 pandemic as the most recent example. So the question really is, it's not quite clear at this point, will the COVID pandemic develop as similar to other plagues or differently? We just don't know. So SARS-CoV-2 is a very complex virus. It's a very big virus, 30 kilobase based genomic RNA. It's the largest plus-strand RNA virus. The virus encodes 4 structural proteins, including the spike protein, which you probably know all about, and 25 non-structural proteins, which the virus uses for its replication, for inhibiting and altering the cellular metabolism and gene expression to facilitate its propagation, and to inhibit the defense mechanisms of the cell, the innate and adaptive immune response. So there are plenty of targets for mutations that can damage the host defenses and improve virus replication and infectivity. And that's why we probably don't know exactly what this pandemic will bring compared to the past pandemics. So I think it's not clear at all at the moment. So the virus infects basically every major organ. The lung is the first station of infection, and then the heart, the brain, the eyes, the nose, liver, kidneys, and so on and so forth. And particularly the brain is particularly worrisome because there are many systemic diseases like inflammation, stroke. There could be peripheral nervous system involvement like anosmia and loss of taste, meningitis, and immune-mediated diseases such as Guillain-Barré syndrome, which is a paralysis condition. And this was actually very early recognized as being a late-stage complication of COVID-19. And it is here in 2020. And there are many autoimmune-related conditions as a late consequence of infection, like Guillain-Barré, progressive paralysis, the purpura, really antibodies against platelets, which leads to bleeding, pulmonary embolism, stroke, multisystem inflammatory syndrome in children, like Kawasaki-like disease, type 1 diabetes, myasthenia gravis, Graves' disease, and the list goes on. What is interesting is long COVID, so these complications are poorly or not correlated with the severity of the initial infection. So if you have a very mild infection, you still can get these late stage complications. And they're not correlated with the risk factors. And the vaccination protects only partially. So we just don't understand all these conditions. That's really what I want to emphasize here. So what is mystery for us and for many was you get infected with the virus and you get a very high titer of virus in the first week or 10 days after infection. And then virus disappears. You can't see it there. But you may remain PCR positive for a long, long time, for weeks or months. And even if people who are discharged from a hospital without virus, they can come back, become PCR positive later or stay for a long time PCR positive. So this one possibility to explain it was maybe SARS-CoV-2 sequences can be integrated into the host genome and become a stable part of the patient. That's one possibility. So we came up with this hypothesis. Could SARS-CoV-2 RNA be reverse transcribed and retro-integrate into the human genome? And could this explain the PCR positivity long after infectious virus is not detectable anymore? That was the hypothesis, and the basis for this hypothesis was twofold. It was my background in retroviruses and unlike an island off the coast of Maine. So let me just briefly give this background to you. The island off the coast of Maine, I like to go there together with my colleague and friend, Rick Young from the Whitehead Institute. It takes us two hours to get there, and then we arrive there at the very beautiful island. Nathan's Island, it's our favorite island. And that was about 2 years ago in June. We did this, we got to this very nice sunset. We had a good bottle of wine. The lockdown was ongoing, and we talked the whole evening about COVID, SARS-CoV-2 biology, and came up with this hypothesis. Now, retrovirus infection. This is a cycle of retrovirus infection, and really a obligatory step in the replication cycle is the integration of the virus as a provirus into the cell's genome. And that is really important for the virus. Coronavirus is a positive strand RNA virus, and there's no reverse transcription in its life cycle. It has a genomic RNA and then subgenomic RNAs, which I come to in more detail. So this is the genomic RNA, 30 kilobase pairs, and these are the subgenomic RNAs, which are the 3' end, and they encode the structural proteins of the virus, like the spike protein or the nucleocapsid protein, which we talk about. They are much more abundant than the genomic RNA. And this is the replication cycle of the virus. The blue is the plus strand, the protein coding strand, and the gray is the minus strand, which serves as a template for synthesizing the plus strand using the the viral polymerase. Now you have the full-length genomic RNA, but you have also the subgenomic RNA, which is made by jumping, by discontinuous transcription from this leader to internal leaders. And they form these subgenomic RNAs, which encode, as I said, the structural proteins of the virus. And they're very important for my talk. So genomic integration occurs, as I will show you, by line 1 retroposition. Now, what is line 1? Line 1 is a transposon. It encodes 2 open reading frames, and 1 of 2 encodes the reverse transcriptase and the endonuclease. And these line 1s make about 70% of the human genome. The human genome is littered with these, and about 100 copies are active. They're active in human embryos and in brain tissue, but normally are repressed in somatic tissues. And they're activated by cellular stress, by cancer, by aging, by viral infection. I will come to this. So this is the mechanism how line 1 integrates. And that's really a bit more in detail, but this is the line 1 and makes a cut in the genome through the endonuclease. And then on the polyase side, the first DNA strand is primed, right? The DNA strand, and then a double strand is made and it integrates at the cut site. The footprints of line 1 mediated reposition is target site duplication. Very important, 7 to 20 base pairs. And a line 1 endonuclease recognition sequence present at the target and a poly-A tile. Once you have those, once you see in any integration these criteria, you know integration has occurred by line 1 mediated integration. So these are the 3 people whose work I'm talking about. Ligo Zhang, who joined my laboratory in 2020 in July, just after we went to this island. Alexa Richards, who is the virologist in my laboratory. She did all the infections I talk about. And Poonam Bisht, who joined the lab just more recently, this project. So the key issues we confronted were direct evidence for genomic integration of corona sequences, criteria for expression of integrated sequences, and potential relevance for disease such as long COVID. Now I'm going to talk about the first points, and I have some data, and this third point of all speculation. So coming back to retroviral replication, as a criterion for genomic integration of the retrovirus, the viral sequences are fused to cellular sequences. Now this is a provirus in the genome. This is a provirus here. It is fused to cellular sequences which flank the provirus. So this was a criterion for integration. So we looked for integration, looking for these type of fused sequences by 3 methods: by nanopore long read sequencing, by whole genome sequencing, and by target enrichment sequencing, which is also called tagmentation. This is the most sensitive method. It's about 200-fold more sensitive than these other 2. Anyway, three independent methods were used, and all 3 approaches showed unambiguously that SARS-CoV-2 sequences can integrate into the genome of infected cells. And these sequences will be a stable part of the infected cell as long as the cell survives. So as a side note, in most experiments in the literature, only one of the 3 methods has been used to detect rare integrants. We use all 3 because we want to be sure, right? And so we were very, very confident that indeed the viral sequences can integrate in infected cells growing in culture. So we find 2 types of integration. One is the one I talked about, line 1 mediated retroposition with a 7 to 20 base pair target site duplication and the presence of line 1 internuclease consensus sequences. It's about 2/3 of the integrations occur by this method. But 1/3, we find integration of a cytoplasmic reverse transcribed DNA copied by homologous or non-homologous integration. Now this, I don't want to go in details here, but this is exactly what has been found more than 20 years ago by Zinkernagel in the mouse virus, LCMVR virus, which is also plus strand RNA virus infecting mouse cells. So there's a different mechanism. It could be mediated by endogenous retrovirus or by line 1, we don't know. So line 1 is expressed at the low level. How could it promote integration in SARS-CoV-2? Well, line 1 is activated in aging, in cancer, and by cell stress such as virus infection, cytokine exposure. Now, I want to point out, aging, cancer are risk factors for the disease, for COVID-19. Cell stress and cytokine storm, so cytokine overexpression, is a consequence of virus infection. So my first summary would be, SARS-CoV-2 subgenomic RNA can be retrointegrated into the genome of cultured cells by a line 1 mediated mechanism. The fraction of cells with integrated copies is very low, 1 in 1,000 to 1 in 10,000 of infected cells go in culture. And line 1 expression is induced by cell stress, like aging, cancer, and virus infection and cytokine exposure. So 2 key questions come up. Is there genomic integration of SARS-CoV-2 in patients? And second, if there is, can integrated sequences be expressed? So these are the 2 key questions which I want to now briefly address. The criteria for integration was the finding chimeric sequences, viral sequences fused to cellular sequences. Now, this is schematically what a virus-infected cell looks like where the virus replicates. 80% of the RNA in infected cells is viral, because a virus has a mechanism to degrade the cellular RNA. So it's a very abundant plus-strand RNA, so genomic and subgenomic. And looking here in these plus strands for chimeric sequences, this really leads to artifacts. Doing sequencing, the reverse transcriptase which you use jumps between templates and you can't use that criterion. But what we realize is when sequences integrate, they integrate either the same direction as a human gene or in the opposite direction. It should be 50-50. And indeed, when we look at our sequences, we find about the same fraction of integrants in the same direction as the gene as in the opposite direction. So this gives us a very important prediction, which I want to emphasize. 50% of chimeric reads from integrated copies should contain negative viral strands. Please remember, this is a very important prediction. But of course, you could argue negative strand viral RNA is also made in a cell which replicates virus. I said this before. We looked at this, and we looked at published data as well as our own data, and we found that it's very, very low. It's between 1 in 1,000 to 1 in 10,000 RNA copies. So the second important realization was in acutely infected cells which replicate the virus, the ratio of minus to plus strand viral RNA is less than 1 in 1,000. So these are 2 really important criteria to understand what I want to tell you now. The prediction of 50% chimeric reads from negative strands, and the negative strands are very, very rare when the virus replicates. So we looked at patient samples, published patients. I give you one example, one of many. And this would be a paper from last year from this group. And here's different patients plotted, and here's a fraction of negative strand viral RNA. And each dot is a lung sample, a lung biopsy of a lung autopsy sample from a patient. So for example, the patient number 5, there were 4 of these biopsies of these samples were highly positive, 2 were negative. And this one, all 5 were positive. That was it. And important is up to 50% of negative strand RNA is found. Up to 50% means, if you remember my prediction, all of the viral reads come from integrated viral sequences. Now I should point out that the total concentration of viral RNA is much lower than in an infected cell. And some of these reads were good enough. You could look for chimeras. You could look for negative strand RNA fused to cellular RNA. If you did find them, as patient number 9, right? Here, these sequences were from the negative strand RNA. So negative strand transcripts are orders of magnitude higher in patient than in the productively infected cells. This is totally consistent with genomic integration of SARS-CoV-2 sequences, if you follow my arguments from before, and expression of integrated sequences. Up to 100% of viral transcripts seem to come from integrated sequences. Now, genomic integration, we can't see directly. Our methods are not sensitive enough. So it's indirect. Look at the RNA. So we have many more of patient data which give you this type of pattern. So my second summary would be SARS-CoV-2 sequences integrate into the genome and can be transcribed in patient-derived tissues. Most, if not all, viral transcripts may originate from integrants. And this could explain our initial hypothesis that many patients are PCR positive in the absence of detectable infectious virus. So our paper met strong criticism and sometimes open hostility. I mean, the classical virologists hated it. So this is a paper that was published a year ago. And we got challenges from people as comments to the PNS and others, and we had to correct those. But we feel very, very confident that indeed virus can integrate into infected cells. So what are the possible consequences for the course of disease? That's really the key issue. And the open questions are, what is the prevalence of SARS-CoV-2 retrointegration in patients? It may be only a very small fraction of cells in a given tissue, maybe not detectable by current methods. But with more than 500 million people infected, even a small fraction will be of significant clinical relevance. So we don't know the answers for this. But what could be the consequence of viral integration? I think the key question is, do integrated SARS-CoV-2 sequences really are translated, express viral or neoantigens, depending on the open reading frame? We don't have any evidence for this. If so, can these antigens induce an immune response to provide continuous protection? Or, alternative, could they elicit an autoimmune response as a late stage complication as we see? So these are open questions, very important questions, very difficult to get to the ground. So I will not really, it's only speculation. But the loaded question is, can vaccine RNA also integrate into the genome and potentially induce an autoimmune response, as we see sometimes in rare instances? And the vaccine question, vaccination, of course, very loaded question. It's as controversial as the election. As you can see here, it's a matter of freedom and of knowledge. This guy here knows more than scientists. And it has been claimed that some of these vaccines can convert you to a monkey. So the question really was, does SARS-CoV-2 RNA, when transfected into cells, integrate into the genome? What is the evidence for this? Now, a paper came out a few weeks ago from a Swedish group. They used vaccine RNA from BioNTech, Pfizer, and found that when they put this into cells growing culture in liver cells, they found in the cytoplasm a copy of spike DNA, and they concluded that was a product of reverse transcription. They suggested that. They didn't have any evidence for genomic integration. They didn't look at. So it was only a DNA copy in the cytoplasm. Now, people have raised objections to this paper because some things were not clearly defined, could be an artifact there. So I don't want to go into this. But that raised the question. It raised the question, is there genomic integration? Because it was only a cytoplasmic copy of the DNA. So that's what we wanted to test. And we made 3 types of RNA: nucleocapsid RNA with polyadenylated, nucleocapsid RNA polyadenylated with a cap, so to allow translation of the nucleocapsid, and capped nucleocapsid where the RNA was modified by introducing a methyl C and a pseudo U, which is exactly in the vaccine RNA. These modifications are done to reduce the toxicity of the RNA and the innate immune response. So we tested this, and this is basically the subgenomic RNA. We tested this and transfected this into RNA. And first of all, we saw that the cap leads to nucleocapsid expression in red here with an antibody. But if you don't have a cap, you don't get expression. No, we tested two different cell types, either 293 cells, which is cell line which is susceptible to coronavirus, and we can use it as a good, easy transfectable cell, which either expressed L1, line 1, so we transfected line 1, or did not express. When we overexpress line 1, we find almost 100 integrations. If the RNA was modified as in the vaccine, it was only a third of this, or less than a third. And there was no nucleocapsid, no translation, that was even much less. So the conclusion from this was all integrates have footprints of L1 rate of position. Importantly, no integration was found if you don't overexpress line 1. And the results suggest that nucleocapsid can facilitate retro integrations, because only with the cap, when you have translation, you get higher integrations. We looked also in a different cell, in smooth muscle cells made from ES cells, and they were also transfected. And after 3 days, we looked at this after degrading the RNase A and beta-glucan. We find that again with L1 overexpression, line 1 overexpression, we find more integration in the presence of a reverse transcriptase inhibitor, AZT, than with the inhibitor. So it was dependent on reverse transcriptase, but no integration when you don't overexpress line 1. So you could argue this is rather artificial. Line 1 overexpression is, of course, not what normally happens. It's artificial. It was a positive control. So line 1 is expressed at a low level. How could it promote integration of SARS-CoV-2? Well, by cell stress, such as virus infection, cytokine exposure, double-stranded RNA, or oxidative stress. So we use double-stranded RNA as a stress. And I give the preliminary, the summary of our preliminary data in cells which were stressed with RNA. We detected nucleocapsid DNA in the cytoplasm. But genomic integration cannot be excluded. It was just how the assay was done. We cannot exclude. It was clearly DNA there. But we also detected DNA which was integrated because it had cellular flanks fused to the viral RNA, but no target site duplication. So the integration was either by homologous or non-homologous integration, which has been seen more than 20 years ago by Zinkernagel in a mouse virus, LCMV virus, infecting mouse cells. So there's a different mechanism that can integrate. So this is very preliminary, and we don't know this. So this is the model we have. So we can get integration by target site primary reversion by line 1 mediated in integration, right? And from a cytoplasmic copy, DNA copy, by homologous or non-homologous recombination. That's what we believe is happening. We don't know what the mechanism of this could be. Line 1 could be endogenous retrovirus. So the conclusions from this are: transfected nucleocapsid RNA can retrointegrate in line 1 overexpressing cells. Nucleocapsid protein facilitates retrointegration. RNA modification, as done in vaccine, reduces retrointegration. Stress can induce endogenous line 1 expression. And reverse transcribed nucleocapsid CDA can be detected in stressed stimulated cells and could be integrated through homologous or non-homologous recombination. So that's our key conclusions. So if I summarize, all our preliminary evidence is consistent with the vaccine RNA not integrating into the genome. But this is such a hot button issue. We have to be totally sure. So it's very preliminary, these conclusions. So I want to really emphasize this. So the challenges, what are the challenges, the questions? So the hostility from the virology community, it really affects badly my ability to get grants because these people are the reviewers. Reintegration events, how could we directly identify those in patients? That's a big challenge. Neoantigen expression, so expression, protein expression from the integrations. Are there preferred integration sites? All these things we're very interested, very difficult to get after. The potential immune responses, such as activated CD8 T cells, they could be the ones who mediate any autoimmune late response. It probably doesn't exist in patient databases. So that's a challenge. And can cells with integrated copies stimulate autoimmunity? I mean, all these are important questions and will be not easy to simulate. Let me come to the final question. That will be just the last 2 minutes or so. Namely, why are bats, no, I'm really switching a little bit of topic now. Why are bats resistant to SARS-CoV-2 infection? Now, bats are thought to be the source of these viruses, not only of SARS-CoV-2, but also of MERS and of other very dangerous viruses. So bats, there are about 1,400 species of bats. It's a very rich species. They're long-lived. They live about 10-fold longer than humans, considering their weight. They have a unique metabolism, up to 1,400 heart speeds when they're in flight. They can only fly for 1 hour, then they have to rest. And they're reservoir for tolerant to many highly pathogenic viruses such as SARS, Ebola, rabies, Marburg virus, Lyssa, Nipah, and so on and so forth. And they carry many endogenous viruses like retroviruses, coronaviruses, filoviruses, parvoviruses. And they, of course, as I said before, they're the source of these jumping between species, these viruses. And SARS-CoV-2 is thought to come from bats. So the immune response of bats is of great interest. It could be too strong or too weak. And both of those could help recover and recovering from virus infection. I mean, strong immune response can lead to really bad side effects, and many of the we see in humans and in patients is the symptoms is thought to be due to a strong immune response, but could be also too weak and then the response, and then you get death. So in bats, how bats deal with this is not clear. So we were very interested to find this out. So what is the cell source? The cell source we're using is bat iPS cells, and they've been generated by Thomas Zwacker from Mount Sinai. We collaborated with him. And these cells are quite weird. They're filled with small vesicles like here. Never seen that. And are there footprints of viruses? They do not belong to any known virus family. And the cells are really also weird. They have double-stranded RNA, which is, of course, in replication of viruses. You see this here. They have endogenous retroviruses here, and they have pan-corona antibody can detect this. So it's very, very strange, these cells. So we were interested to see, can they be infected by SARS-CoV-2? And we infected those, and no infection. So the question was, why was there no infection? Was it because the receptor was missing, or was it further downstream? So we expressed the receptor in these cells, the human AC2 receptor, and then they can get infected as a GFP-labeled virus. And you can see here in the Vero cells, which are producer cells, they can be very well infected. And also Delta virus can very efficiently, you can see much more efficiently than this other virus, can efficiently infect these cells, these bat iPS cells. And we know the cells, infected cells, produce virus. We can titer the virus. And you see a plaque under the scope, and that goes down after 5 days. So our conclusions are, and this is really all I want to say, it's very preliminary, the bat cells are can be efficiently infected with SARS-CoV-2 if they express the human AC2 receptor. And the toxicity to virus infection is probably less than in human cells. So the questions, which may be interesting to follow, is toxicity of SARS-CoV-2 for bat cells different from that in human cells? We don't know. What is the immune response of these cells? And is there genomic integration? So we believe studying the bat cells, it's really started this by lab, this can give us some hints to understand more about the biology of the viruses. So I want to finish here and acknowledge the people who did this work. I talked about Ligo, Alexia, and Poonam, who did all these experiments I talked about. We collaborated with Eric Young at the Whitehead, Inma Barasa from Whitehead Computing, Thomas Zwacker on the bud cells, and Steve Hughes from NCI. And with that, I will stop by sharing and be open for questions.
Thank you so much, Rudolf. Thank you for that wonderful presentation. And I'll kick off the Q&A portion of the discussion. And it looks like we have a lot of questions that have actually come in, but maybe I'll start by asking a question. Rudolf, have you looked at all at the different variants and how any of the variants might differ in their ability to integrate into the genome?
No, we looked, you've got the Omicron variant, but we haven't really quantitated this. And we looked at the Delta variant in bud cells, but again, we can't compare that.
Right. Okay. All right. Well, heading to some of the audience questions, one of the questions is, how might genomic integration of SARS-CoV-2 and genetic sequencing be used to predict the probability of contracting disease and help kind of influence healthcare costs or drive down costs and reduce inequity?
Well, the integration is sequencing is very inefficient. It's not sensitive enough to see integration. But you could look at RNA as we did. But the RNA will come only once the acute, become only relevant once the acute infection is over, I assume. And then it could be a good criterion to understand what's going on. That's an interesting idea. It hasn't been used at this point.
The next question relates, I think, to some of the fundamental biology of SARS-CoV-2, but what cellular factors regulate SARS-CoV-2 infection either positively or negatively? And are there any factors that you've identified that might influence genomic integration, I think, as a kind of a corollary?
So the infected cell mounts an innate immune response, which really tries to damage the virus factory to produce viral proteins and so on and so forth. The virus counters this and fights that part. So it's a battle between the cellular defenses and the virus trying to overcome the defenses. And in SARS-CoV-2 infection, the virus wins most of the time. But of course, the cellular defenses are very important, particularly then the immune system later kicks in and produces interferon, and that will limit the viral spread. So that's very important. So mutations, which affect, so interference is very important, and it has been shown that people who have a defective immune interferon response are much more sensitive to SARS-CoV-2. So some people who have a genetic defect in the immune response are more sensitive. So there are a couple of really good candidate genes, cellular genes, which affect the virus replication.
Okay, and then on the topic of integration, can genomic integration of SARS-CoV-2 result in mutations that are linked to cancer or other diseases? And can these mutations affect the germline, do you think?
So, okay, there's a complex question. So first of all, I don't believe so, because what integrates has no promoter. So if a retrovirus integrates, that can cause cancer. That's how retrovirus, leukemia virus, and other viruses were discovered. So that's really they can turn on an adjacent gene. These cannot turn on. They can cause a mutation, sure, by integration, but the mutation would be certainly only a recessive, would be only one copy of the gene. So very unlikely it could activate an oncogene or inactivate two copies of a tumor suppressor gene. So I think it's very, very unlikely. And the other question was, no, that was what it was, right?
I guess the next question is, how does SARS-CoV-2 genomic integration help in finding new targets for vaccination, or do you think there are implications for vaccination with genomic integration?
So one thing we know is that that the process of integration really requires the action of reverse transcriptase. So reverse transcriptase either coming from line 1 or from a retrovirus. Right? So anti-retrovirus, anti-RT, reverse transcriptase drugs like AZT, which is used for treat HIV patients, should, and we have shown this, does reduce integration. So this could be, if it turns out that integration has relevance for the disease, that could be one way to prevent or to diminish integration if you take such a drug soon after infection.
Okay. And the next question is from Brian Mayarella from UC Berkeley, who asks, what was the size of viral integrants that you detected? Do they correspond to full-length viral proteins?
No, not at all. The biggest piece was exactly the nuclear capsid. It's about 1,600 base pairs. It's 5% of the genomic RNA. So it's almost unthinkable that infectious virus could be, and the whole full-length genome could integrate. To my opinion, it's totally impossible. So this was only one we got. Most of the other fragments are shorter. Anyway, that was the longest one, 5%.
Okay. And then the next question is from Monica Concepcion, who asks, can you discuss what you know about superimmunity? Or I guess, are there implications for patients that might be protected for SARS-CoV-2 or resistant for SARS-CoV-2 infection?
Well, resistance would be if you who is really resistant to the virus? If you have the right immune system, you probably do better. If you don't have these risk factors, which we know is obesity and immune compromised or diabetes, no, these things. That's if you don't have this, you're more easy to handle the virus. A real resistance gene, to my opinion, has not been identified. A gene which gives you resistance directly. So it's more not having these risk factors that gives you resistance.
Okay. And Poria Hosseini asks, even without viral DNA in the cytosol or in the nucleus or without integration, is there a risk for autoimmune responses since the human cells are producing a foreign protein?
Yeah, sure. I mean, but you know, it's just RNA, it's just a virus infection. The cells in general die. So this exposure to this antigen, viral antigen or whatever protein, would be short-lived. If it integrates, and there's evidence that cells in the nose, for example, a paper from the Broad Institute, they are long-lived cells which produce negative strand RNA. They're long-lived. They don't produce virus. So there could be some cells in the body which are long-lived but carry viral sequences. We don't know that, but this is suggestive. It's totally consistent with the hypothesis. So for the autoimmune disease or for autoimmune symptoms to occur, you probably need a longer time exposure. And that, I believe, will be not easy to achieve just with transient viral infection by RNA.
Okay. And another audience member asks, why does the virus research community object to your results? Do they take issue with your experimental systems, your results? What is it specifically?
Well, they hate this because it's a novel idea. So that's something which always is a problem. And Rolf Singer, they told me the same thing about when he published this paper 20 years ago, was also a lot of resistance. But so the history of this paper was that initially we did not think there was this artifact of which I mentioned briefly, of plus strand RNA looking for chimeras. It gives you artifacts in the sequencing, and we were claiming this in a preprint in December of 2020 that we had evidence for integration, and people rightly criticized us for this. So that was a wrong paper, was wrong. We were forced to put this on bioarchives. It was a mistake, and we got bad criticism. It was the biggest crisis in my career. And that's why we went to this extraordinary effort to look for integration by these three different methods, as I said, to convince ourselves there was direct evidence for integration, not this indirect evidence from looking at plus strand, plus virus strand fused to a viral RNA, which could be an artifact. So I think these two things coming together with people are critical.
And Edward Kukla asks a little bit more about long COVID and asks, what is your impression as to the cause? Is everyone who is SARS-CoV-2 infected carrying the COVID virus in their genome or just some?
Well, I can't answer this question. It's all right, purely speculation. I think what we know is long COVID, you can't predict it. Even people who are very mildly infected or people who are very deeply, badly infected, they have a chance, probably a similar chance to get long COVID. So I pointed this out. So we really don't know. We are just ignorant about what causes long COVID. And that's, I think, makes it such a, in a way, frightening and also interesting issue. Right? It's just, that's not clear to me.
Okay. And Erwin Jungreis asks about some differences between different viral components that might be integrating. So have you tested whether the nucleocapsid protein could be integrated, but the vaccines have spike protein rather than nucleocapsid? Have you tested nucleocapsid versus spike protein integration?
We concentrate only on nucleocapsid. All our data were nucleocapsid because the nucleocapsid subgenomic RNA is the most abundant RNA in the virus infected cell. So there's a highest chance to get integrated. I think it's very important to look now at the vaccine, at the spike RNA. And we haven't done this yet, but we're planning to do that.
Okay. Christopher Hugg asks, might therapeutic editing, genomic or ADAR, affect infectivity of the virus? Can this drive viral evolution or therapy?
Yes, it could be. It possibly can. And the virus can evolve, as we know from these variants coming up. It's quite, to my opinion, it's very, very interesting to see how new viral variants come up that seem to be not as causing such serious disease, but then they can replicate better. But as I try to point out, the virus has 25 genes which can affect the cellular response and the defense mechanism. There are many, many targets for mutations. So we cannot predict whether a new variant could also cause, again, more serious disease. So we don't know.
Okay. And Shahab Sarmashki asks, aside from some of the work that you mentioned on retrotranscription HIV drugs that might be useful after infection, are there other implications for therapeutic avenues that are brought up by your research finding that there is genomic integration?
Not one if I can think of, to be honest. So that's really, that's one we show. It's dependent on reverse transcriptase activity. So we can inhibit this. But it would be only justified once we have evidence that integration really is important for these late stage effects.
Okay. And then someone in the audience asks, has similar integration been seen for other coronaviruses, such as those that cause common colds? If not, what do you think the difference might be?
We looked at other RNA viruses, like Zika virus, like dengue virus, like flu virus, looked for integration in either our own data, because we work with Zika virus, also, or in published data, and we have not found it. So this could be two reasons for this. Either it's really clear it's not there, or these infections with these viruses are so toxic that we can't wait long enough. In the samples we looked at, we couldn't wait long enough to find integration. So in the corona work we do, the cells die after 2 or 3 days. So we don't have much time to wait. With the vaccine RNA, we hopefully can keep vaccine RNA transfection to look for integration. We hope we can look for longer time and to accumulate viral integration. So we hope that, but we don't know. We don't have any evidence at this point.
And Doug Gray asks, can SARS-CoV-2 DNA sequences be detected in the genomes of bats?
Well, good question. We're looking at this. So that's one thing we'd love to know. We would like to know how bats deal with that infection. Can they either integrate efficiently, that's for that part of their defense mechanism, or they can exclude it? We have no idea.
Okay, and I guess along similar lines, could bats be resistant to SARS-CoV-2 because the virus they carry is not the same as the one that's circulating in the human population, or there are differences that might have occurred?
It's certainly true, because the human SARS-CoV-2 cannot infect bat cells unless you introduce the receptor, right? So we have shown that. That's quite clear. So it's not a block downstream of fusion of the virus to the cell. So that's not. Otherwise, we don't know. But clearly, the cells do have double-stranded RNA in the cytoplasm, and they seem to have antigens which are detectable by a pan-corona antibody, and lots of endogenous retroviral RNA. Now, is this active virus replication, or what is it? We don't know. It was done. Some group did very careful tests for looking for infectious virus. They're absolutely unsuccessful to find any infectious virus produced by these cells. So that's where we say we just don't know enough about this. It's very interesting cells, but we have to learn quite a lot.
Okay. And there's a question about cell types for which the virus may have a preference for integrating. So someone asks, can the virus integrate specifically in germ cells? But I guess more broadly, are there certain cell types that you've identified or that you've found where the virus may be more prone to integration?
So we're looking at this. So we have the data we have from patients for lung, of course. Lung is the most data of this. But then we found from our own, we looked at many different cell types produced from human embryonic stem cells. So, and there we know that liver cells are very affectable. Liver cells and smooth muscle cells are. Neurons are not. Heart cells are very little infectable. Endothelial cells are not infectable at all. So clearly there's quite a spectrum of the tropism of this virus. Now, question to the germline. Germline integration is very interesting. Now we find that Ebola virus sequences are in the human germline. So at some point, this virus found its way into the germline, probably very inefficiently, and got integrated. And other viruses are in the human germline. So it can occur, but it's I think, very unlikely in a patient that this would infect the germline. And anyway, it would never be infectious virus. It would be only a little piece of the virus.
Okay. And then an audience member asks, are the quantitative proportions of subgenomic transcripts generated from an integrated copy of the virus the same or different than those generated during acute cellular infection?
So the subgenomic RNAs from acute infection are made by the viral polymerase, by viral RNA polymerase. If the sequences are integrated, there's no viral RNA polymerase. These cells are free of virus. So they are transcribed by the cellular POL2 enzyme. And so it's a very different mechanism. There's no subgenomic RNA. It will be fused chimeric RNA produced by the POL2.
Okay. Well, thank you, Rudolf, for that wonderful discussion. Thank you for answering all those questions. That concludes today's presentation. Thank you all for joining us today. And a special thanks to Rudolf for sharing your work with us. And goodbye, everybody.